Hello, and welcome back to Human Evolution. Today I want to talk a little bit about tracking genes through families, and then also special cases where you have genes that are on the sex chromosomes, and that is going to look a little bit different than the genes that are on the autosomal chromosomes, the ones that we've been dealing with up to this point. And then I also want to talk about the fact that the majority of your traits are actually what we call polygenic, and I want to get into what that means and what that looks like. So, first things, why are there more males with color blindness than females? Well, it turns out that color blindness is a sex-linked trait. And it might be tempting to say that there are more males that are colorblind because it's on the Y chromosome, but if that were true, then there would be no females who are colorblind, and there are colorblind females. There are more males, but there are some colorblind females, which means it has to be uh, a sex-linked trait, number one, and it also has to be on the X chromosome. It turns out that color blindness is a recessive gene, right? And it's usually going to be paired with a dominant version of that gene that's normal because the wild type is normal. Yeah. So B is the answer to that. And let's take a look at it. So if you have father, right, has two potential sex chromosomes that he can give to the baby. He can either give an X or he can give a Y. Mom can give an X, right, or the other X, and these may be different because they may have different things on them. And we've talked about this a lot, so this is just a review right here because it's going to help us figure out how to read these pedigrees that I'm going to show you in a second. But here's your next question. A person that's a carrier of a genetic disorder, thinking back to what you were just looking at, what is the likelihood that they'll pass on the defective gene? Well, if you have two alleles, right? If you have two chromosomes for every pair, one of those chromosomes goes into the gamete, then it's a 50-50 shot. And so when we start looking at, say, affected things, like the actual traits, if you have an unaffected father, right, he's always going to give the unaffected X to the girls, right? If you have an affected father, by the same token, then he'll always give the affected X chromosome to the girls because he's just got the one, and then boys get the whole Y situation. Anything that's on the Y chromosome will only show up in boys and will be passed through, obviously, the male lineage, okay? Now then we've got mom, and she's got two. You can get one of these or one of these, okay? And so what that results in the children is here for the boy, right? Let's say this boy gets the unaffected X from mom, he's got the Y, he's unaffected. This boy, on the other hand, got the affected from mom, right, and he's got the Y, so he is affected. The girls also have two possibilities. They'll get the unaffected X from dad, and then from mom, they're going to get either the affected, right, so this daughter's a carrier, or they'll get the unaffected, which means that the daughter is completely unaffected and cannot pass on the gene. The mother was a carrier and she can pass it on. This heterozygous daughter is also a carrier and she can pass it on. Okay. And here's just an example with a very famous case, and that is hemophilia. When the mother carries the hemophilia gene and the father is unaffected, it's the picture that you just saw. It's 50-50. Okay. When the father has hemophilia and the mother is unaffected, none of the sons will be affected and all of the daughters will be carriers, okay? And that makes sense, it's what we just saw, but here's a real life example, okay? So, in cats, gene E produces yellow fur and gene B produces black fur. A cat that inherits both of these genes has patches of yellow and black fur and is known as calico, right? Sometimes we call that yellow-orange, right? That's also called orange. The alleles for black and or yellow are located on the X chromosome. The cross XB for black, Y, crossed with XB, XE, right, which would be the orange, is this Punnett square here. The yellow male offspring are represented by 1, 2, 3, or 4. There will be maybe more than one answer to this question, but maybe not. Well, if we look at it, this is XB, we've got XB, okay? This male is XBY, and this male is XEY. So what we've got here, black kitty, right? Black female, black male. calico, right? And female, and orange male. By the way, calicos are all females, as we've discussed, I think, before, but if we haven't, calicos are all female because of this whole like 2X thing. Okay, and we'll either talk about that later or we already have talked about that. I can't remember, but it'll come up again. Okay, so when we're tracking genes, this is a pedigree, right? And so what we do is we look at all of the people that are in the family, who's affected by whatever we're looking for and who isn't. Females are always going to be circles and males are always going to be squares, so remember that. So here we're looking at hemophilia, with the parents being Queen Victoria and Prince Albert, right? So Queen Victoria is a carrier. Yeah. So the question becomes, how many of her children will be affected or, be carriers, depending on if they're males or females. And so when you look at her kids, right? So Edward VII clearly got her unaffected gene. Okay. Victoria got her unaffected gene, right? Alice got her affected gene, but then she got that unaffected gene from dad, and so she's a carrier. Okay. Alfred, Arthur are just like Edward. Leopold, on the other hand, right, he got her affected chromosome, right, because he just gets the one, so he got that from mom. So Leopold is a hemophiliac, right? His daughter Alice is going to be a carrier no matter what. Okay, and that's because she'll always get that affected gene, right? All of his daughters will be carriers. And then Helena, and I don't know who that is, are both normal females, and then Beatrice is also a carrier. Okay, and it goes back to the idea that if mom is a carrier, there's that 50-50 shot that you're going to have hemophilia. And so you can see and you can track through this pedigree who's got hemophilia and who doesn't, with that idea that you've got the 2 X's for mom and the 1 X for dad. And this is, guys, this is going to be true for traits, whether they're sex-linked or not. You have pedigrees for autosomal traits too, and we're going to look at that as well. But we just happen to be talking about sex-linked to kind of complete that conversation about how genes are passed on, right? So the autosomals, you have 2 copies from each parent. You have 1 copy from each parent, but each parent has 2 copies of the same gene, maybe different alleles. In the sex-linked, the reason it's different is because you have that XY with dad. And so we're just kind of finishing that conversation off of the notes that we had before, okay? But pedigrees can be used to track autosomal traits and sex-linked traits. And here's just an example of another pedigree. And this one I like because it has monozygotic and dizygotic twins. So these are identical twins. These are fraternal twins. You can see that they're just siblings born at the same time, and these guys came from the same X. Sometimes we don't know about people, especially if we go back really far in the past, and so sometimes we'll have an unknown or unspecified sex. We read the generations by the Roman numerals on the side, and then we have, of course, the females and the males, and then usually the shaded is the affected, but that is really going to depend on the pedigree. It really does not matter. I mean, it depends on the pedigree. Okay, so using that, what is individual 2,4's genotype? Well, to do this, we're going to look at the Roman numerals and then the numbered individuals. Okay, so 2, 4 right here, right? So you might be tempted to say that, oh, I don't know because I don't know what the trait is and blah, 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 blah. Well, that's all well and good. But the fact is that this child is different than these 2 parents, okay? So what this means, number 1, is that this is an autosomal recessive pedigree, okay? And so the gene that we're looking at, most likely it's these dark shaded ones. Remember I told you the shaded ones are typically the affected ones, but in this case you can see that that's true. You have these 2 parents who have 2 normal and 2 affected, right? So these guys are homozygous recessive. This girl and this guy, this female is homozygous recessive as well because she's also a carrier. But since parents here both are normal, that means they both have to be heterozygotes. So 4 is big A, little a. Now, I just want to call your attention to the fact that, we also know that 1 and 2 are both heterozygotes for the same reason. But 1 over here and 2 over here, these guys, right, may in fact be homozygous dominant. We won't know until we see their children, because if you think back to that Punnett square, that ratio of 1 to 2 to 1, it's very possible that 2-1 and 3-2 are actually homozygous dominant, but we don't know. Okay, but we do know that 2-4 is big A, little a. So the pedigree that we just saw is an autosomal recessive we talked about, and this is that. What you can see here, and the dead giveaway, is that you'll have unaffected parents that have affected children, which means they're heterozygote. This is the idea of skipping a generation, by the way, because you can have normal parents that have affected children, right? In an autosomal dominant situation, though, you'll have every generation has the affected trait because it is dominant, okay? And then here's an example of an X-linked recessive. You will see it mostly in boys. It will skip generations. You'll have carrier moms, right? And the carrier moms will marry unaffected males. But the dad is not important to the sons, only to the daughters, okay? Okay, so now we have two more, and I just wanted to show you what do you think this one is? We've got the trait in every generation, so clearly it's a dominant trait. We've got that skipping here, so it's a recessive trait, right? And so again, just looking at that, not 100%, but yes. Here we have PTC tasters and non-tasters. You can actually see that, oh, this is tricky, right? So which one is dominant? If you didn't have this, which one was dominant and which one is recessive? Okay, well, you have these two that don't have the trait, and yet they have a child that has it. Okay, and so whatever this white shaded is going to be the dominant, right? So that means that people can taste the PTC chemical, right? That's a dominant thing. Okay, now interestingly, this homozygous recessive marries a homozygous recessive, which means that you have all of the children are homozygous recessive because there's no option for a dominant gene, and then everything else is normal. So I thought that would be fun for you to see too. Okay, so let's talk a little bit about Mendelian versus polygenic traits. You may have at some point in your life learned that eye color, you had the allele for blue eyes was recessive and the allele for brown eyes is dominant. That is true and not true. First of all, so eye color is actually informed by multiple genes. Now there is a gene that deals with laying down pigment, or whether you actually see the no pigment, right? And so lots of color genes especially have this kind of like pigment, no pigment situation, and I don't mean no pigment in terms of being albino. That's not what I mean, but it's two different scenarios with a particular pigment. That's true for the eye color. So it's not wrong to say that with the blue and the brown thing, but it's also not correct either, because parents, two blue-eyed parents can absolutely have a brown-eyed child, and that child is not adopted. So this is that polygenic situation. All right, and there are lots of human traits that are polygenic. Skin color would be one of them, right? We have this variety. Anytime you see a scale, a gradation of whatever the trait is, that means that it's polygenic, right? And the other thing that you'll notice about these pictures is all of these girls are different heights, more or less. And so height is another polygenic trait. It just means that you have, in this case, there are 3 genes that are dealing with the skin color, right? 3 genes, A, B, and C. And so when you do that Punnett square, right, of all the crosses there, this is what you get. And so you can get all of these different colors, right? And that's pretty fun. There are a lot, like I said, most of the traits are polygenic. Now, the last thing I want to talk about quickly is the molecular clock. Okay. So remember that everybody's got DNA, right? And so we share common ancestors, and it's much better when you're talking about phylogeny to use DNA, because remember, we have things like convergent evolution, we have adaptive radiation, we have pressures that can be very extreme, and so you can get some really different organisms that are really much more closely related than we think by looking at their DNA. And so DNA similarities are a much better way to determine relatedness than anything else, right? And sometimes we can look at just the DNA, sometimes we can look at the actual proteins and their functions and to see how they've changed. So both DNA and proteins can be, and RNA too, actually, can be really helpful, okay? And so when we talk about molecular evidence, we're talking about looking at those changes in nucleotides and amino acids over time, right? So if you have a mutation before species diverge, then you actually have those species sharing that mutation. At some point, speciation occurs and you get a split, and the mutations should accrue separately, right, in those two species. So mutations occur before and after speciation, and that's kind of how we know where speciation events occur. Sequence alignment is what we do. I mean, we just look at the sequences. Okay, ooh, look, that one had a deletion. Yay. Okay, so, oh, look, we have a little swap here. Woo. So when you're looking at these, you can start saying, okay, well, there are 5, 10, 20, whatever mutations that are different between these two groups, right? And so the number of differences can be related to the time, because in related species, mutations occur at a fairly constant rate. Now, obviously, you're going to have groups, bacteria, mutations are going to occur a whole lot faster than they are in, say, humans. But when you kind of get an idea of what those mutation rates are, you can estimate based on what you know about the species. And then you can say, okay, well, in this case that I have here, one mutation occurs every 10 million years one mutation that stays, right, occurs every 10 million years. So if you have 2 mutations, then you know that you're looking at 20 million years, right? So we use different chemicals, right? And by molecules, different molecules, to determine different groups, right? If we have really closely related species, we need something that has a higher rate of mutations so we can see it better. If we're looking for something that is really distantly related, then we need something that has a lower rate of mutations so we can keep track of them and look at the similarities and differences. So for really closely related, we already know that we use mitochondrial DNA. It's how we know that we're about 180 to 200,000 years old as a species. It's how, we know how related we are to, say, Neanderthal. Things like that. But when we start looking at like the domains, for instance, that's a long time ago. There's a lot of stuff that's happened since the divergence between bacteria and then archaea and eukarya, and then between archaea and eukarya. And so for that, we need something that's highly conserved. And that what highly conserved, remember, means that there is a lower mutation rate. Ribosomal RNA is really, really conserved because mutations tend to screw it up. I mean, we already know how ribosomes work. And so there's not a lot of room for changes in that, because you really need the ribosomes to work really well consistently all the time, or else survival is limited, right? So what that means is that it has a lower mutations don't accrue very much in ribosomal RNA, and so we can look at long periods of time there, okay? Now, which of the following is not true when discussing the molecular evidence for evolution? Related organisms share a greater portion of their DNA. The hemoglobin gene is less similar between humans and dogs than between humans and chimpanzees. Only DNA can be examined for establishing evolutionary differences. Molecular evolution progresses more quickly when there's no selection for the gene, or phylogenetic trees can be established using molecular evidence. Well, the thing that's not true is about DNA, right? I mean, remember, I already talked about proteins and said that we can use proteins too to establish evolutionary differences. And here's a very good example. We have this protein that is lysozyme. So lysozyme doesn't necessarily change. So this is where DNA is not helpful. You still are making lysozyme. You still have the lysozyme gene, but sometimes the protein function will change. And so we are looking at two things. One, we're looking at the actual structure of these molecules. And then two, we're looking at what those molecules do, right? So sequence and then function. Lysozyme, you've got it, right? It's in most animals. It helps break down stuff, obviously. Ruminants, okay, have it too in their foregut. And it's what they use to ferment, right? To break down the cellulose that they eat. It's evolved some distinctive changes that will actually protect it from being digested by, say, the hydrochloric acid that's in the stomach too, okay? And so that is different in ruminants than it is in other things, okay? So just as an aside for functions, new genes and proteins are necessary to have new functions occur. So remember that everything that you see out there, all of the diversity, is some change in form or function. And we already know, we've talked about mutations, the sources for new genes, transposition, right, transposal elements, polyploidy, and duplicate genes, gene duplications and duplicate genes, right? Hemoglobin is a really good example of the gene duplication thing. It turns out that the gene families for hemoglobin arose from duplicated genes, okay? And those are called pseudogenes. We're not going to talk about these a lot, but gene duplication is actually very interesting, and it will allow for changes, new functions, things like that. Now, we also know that we can import DNA from other right, genomes, depending on how that is. We know viruses are really good at that. We know that bacteria do that, you know, all the time with each other. So there's that. But for us, more that's going to be a virus kind of thing. But anyway, and we've already talked about that. But the other thing that happens is whole chromosomes can break and they can fuse, right? So if you look at chromosome 1 for humans and chimpanzees, gorillas, and orangutans, our, you know, closest extant relatives, you can see that there's some similarities, except there was an inversion here, right? And so you can see that we had a breakage and an inversion, and so our centromere is in a slightly different place, okay? In chromosome 3, it was the orangutan that had the inversion, right? Not us, okay? And so here, right, you have chimpanzees chromosome 2, well, they actually have chromosome 2 and chromosome 3 right here. They have one extra set. Our chromosome 2 is actually fused, a fusion version of these two, right? And then this centromere disappears. Okay, so that's just another example. And again, organisms that are similar to each other at the molecular level are going to be more closely related than dissimilar organisms, and distantly related organisms accumulate a greater number of evolutionary differences than closely related species, and then phylogenetic relationships are much more sound if they are based on this molecular similarity and difference idea. Okay, so using the molecular record to determine phylogenetic relationships is based on the assumption that 1. Nucleotide sequences don't change over time. Nucleotide sequences change at a fairly constant rate over time. Nucleotide sequences change randomly and erratically over time. Changes in nucleotide sequences change the function of the protein, or all mutations are harmful. Well, obviously not all mutations are harmful because we're here, right? Changes in nucleotide sequences don't have to change the function of the protein. Nucleotide sequences do change randomly, but it's not erratically. It's over a fairly constant rate of time. And obviously they do change because that's mutation. So B is the answer, right? And I just want to give you a really fun example. Pandas, the giant panda, are they raccoons or are they bears, right? This was an interesting argument to try and figure out whether the giant panda was a bear or a raccoon. And I don't mean that they are a bear or a raccoon. Well, they are a bear, but are they in the bear family or are they in the raccoon family? I mean, they clearly look like bears. They climb trees, right? But they don't hibernate and they eat bamboo. They're vegetarians, right? Which is actually similar to a red panda, which is in the raccoon family. So we really had to kind of figure that out based on DNA. It was DNA that allowed us to put pandas in the bear family, right? Although we know that they're more closely related to raccoons than other bears. And so you can see, here's the panda. It's definitely a bear, right? But it is more closely related to raccoons and red pandas, okay? And that's the problem with the idea of phenotypes versus molecular evidence. All right, so if you have any questions, remember there's the video notes board that you can post your questions to, or you can just write them down and ask them in class. All right, thanks for listening. I will see you next time.